Arnon to color and request Sir to present his topic in our pre-conference session. Mr. Arnon, please.
Start my video แล้วถ่ายแชร์อะไรถึงถ่ายแล้วเนี่ยกดแชร์ถามเขาก่อนว่าได้ยินกดแชร์ก่อนแชร์แล้วก็กด full screen ได้เลยถามว่าเขาได้ยินไหมเช็คก่อน
Hello. 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 Can you hear me?
Audible. Yes, sir. You are audible, sir. Hello. You are audible, sir.
น่าจะได้ยิน
Okay. Can I start now? ได้ยินเลย
น่าจะได้ยิน
Okay. Good afternoon, ladies and gentlemen. I'm glad to have you in my presentation today. My name is Anon Chukamne, a PhD student from Divisions of Biomedical Science and Biomedical Engineering, Faculty of Medicine, Prince of Songkhla University, Thailand. So I'm going to talk to you about our research in the topic of the in vitro synergistic activity of phosphomycin-based combinations against carbapenem-resistant capsular pneumonia clinical isolates. So this is the outline for our presentation today. We start with the introductions, material and methods, result and discussions, and finally the conclusion. Why is carbapenem-resistant Enterobacter or CRE important? In 2019, the Center for Disease Control and Prevention, or CDC, reported that carbapenem-resistant Enterobacter or CRE are considered as urgent threats because CRE causes about 13,100 infections per year and about 1,000 deaths per year. And some of the CRE resistant to nearly all antibiotics, leaving more toxic or less effective treatment options. And among them, the predominant strain is the carbapenem-resistant capsular pneumonia or CIKP. So how can Enterobacter resist to carbapenem? In general, there are three carbapenem-resistant mechanisms. First is the enzymatic inactivations by carbapenemase enzyme that can destroy all beta-lactam antibiotics. And the second one is the efflux pump that can transport various antibiotics and other substances out of the bacterial cell. And the third one is the polylase that can decrease outer membrane permeability to the antibiotics. Among them, the production of carbapenemases is the main mechanism of carbapenem resistance, particularly in Enterobacter. The carbapenemases can be hydrolyze the beta-lactam at the beta-lactam ring in the main structures of the drugs, and leading to acid productions and no actions of these antibiotics. It can be classified into three amino classes by differentiations of amino acid sequence, including the class A serine carbapenemases, class B methylo beta-lactamases, and class D oxacillinases. Class B carbapenemases are an important enzyme that widely spread in the CRKP strain. So how can we treat CRE-infected patients? Normally, there are two strategies. First is the monotherapy by using the old antibiotics such as the colistin, dicycline, aminoglycosides, and phosphomycin. However, many studies reported that if we use these antibiotics as a monotherapy, the resistance to these antibiotics might be occur rapidly. And also the development of new effective antibiotics is not easy, take a long time and need the approval in many processes for using in the clinic. So the second one is the second one is the combination therapy, which is such as the phosphomycin-based combination that might solve this problem. And in our study, we use the phosphomycin as the antibiotics-based combination for combating the CRKP isolates because of lower side effects when it's compared with the other drugs of choice. So it leads to our objective that to characterize the carbapenem resistant mechanism in CRKP clinical isolates and to evaluate the synergistic activities of phosphomycin in combination with other antimicrobial agents against CRKP clinical isolates. Next, I'm going to talk about materials and methods, results, and discussion together. This is the workflow of the study. I will begin by the bacterial isolation first. So we got the leftover specimen from a previous research, and this study was approved by the Human Research Ethics Committee of the Faculty of Medicine, Prince of Songkla University. And the process was performed by the rectal throat swab and environmental swab. And each sample was stripped on the McConkey agar supplemented with the imipenem 6 microgram per milliliter and incubated at 37 degrees Celsius for 24 to 48 hours. Then we got 35 capsular pneumonia isolates. For the antimicrobial susceptibility test, all 35 capsular pneumonia isolates were tested the antimicrobial susceptibility by this diffusion methods according to the Clinical Laboratory Standards Institute or CLSI guideline 2016. Briefly, the bacterial culture was adjusted to 0.5 methylene by the sterile normal saline solutions. Then they were swabbed on the Muller-Hinton agar, and the antibiotics discs were placed on this agar plate, and it was incubated at the 37 degrees Celsius for 16 to 18 hours. And these are the antibiotics used in this experiment. So this graph shows the percentage of resistant isolates for 12 antibiotics. We found that all isolates were resistant to ampicillin, followed by the 86% of the isolates were resistant to cefotaxime, ceftazidime, and colistin, and 80% were resistant to piperacillin, tazobactam, and trimethoprim sulfamethoxazole. Interestingly, we found that 21 isolates were resistant to carbapenem, and they were suspected as the CRKP strain. 15 of these isolates were isolated from throat and rectum of the patients. And in addition, there are many studies about the CR in Thailand. The researcher reported that the prevalence of CRKP, which isolated from hospitalized patients during 2001 to 2010 was 0.7%. And the year later, another study, they reported that the prevalence of the carbapenem-based producing enterobacter, which isolated from patients during 2010 to 2011 was 7.7%. And after that, to confirm the carbapenem-resistant isolates, the MIC values of imipenem and meropenem was evaluated in all 35 capsular pneumonia isolates by broth microdilution according to CLSI guideline. Briefly, the bacterial cultures was adjusted to 0.5 methylene by the normal saline solution and diluted 1:100. Then they were pipetted into 96-well plate, which contained the Muller-Hinton broth and 240 concentrations of the drugs. And the Escherichia coli ATTC 25922 was used as a quality control in this experiment. we found that 21 isolates were resistant to imipenem, while 20 isolates were resistant to meropenem. In addition, 21 isolates were classified as the CRKP strain, which concords with the diffusion methods. And we found that the MIC50 and MIC90 of both imipenem and meropenem were 32 and 64 microgram per milliliter, respectively. And then we selected the 21 CRKP isolates for further study. In 21 CRKP isolates, they were investigated the carbapenem resistant mechanism. First, we detected the carbapenem mass production by combining this test. The principles of this method is like this diffusion method, but they compare the zone diameter of this plus inhibitor and this alone. Here we use the meropenem plus phenylboronic acid or PBA, which is an inhibitor of class A carbapenemase enzyme, and the result was interpreted as a positive if the meropenem plus PBA zone diameter is more than meropenem alone, around 5 millimeter than the meropenem alone. And another method is the same with the previous slide, but we use the meropenem plus the dipicolonic acid, which is the inhibitor of class B carbapenemase enzyme. And the last one, we also use we also detected the co-productions of the class A and B carbapenemase enzyme. We found that all isolate didn't produce class A carbapenemase and class B A plus B carbapenemase enzyme. But 18 out of 21 CRKP isolates produced class B carbapenemase enzyme. And the other 3 isolates didn't produce any carbapenemase, but they were resistant to carbapenem. So I will talk about this isolate later. And in addition to the phenotypic detections of carbapenemases, we also detected the carbapenemase encoding genes by PCR method. Firstly, the 21 CRKP isolates were extracted using the DNA using the boiling methods, and the bacterial cultures was heated at 100 degrees Celsius for 10 minutes and put it on ice for 5 minutes and centrifuged at 10,000 RPM for 7 minutes. And the DNA contained in the supernatant. And the amplifications were performed by the PCR using the thermocycler. And we used this primer, which are specific for the BLA-KPC gene in order to detect the class A carbapenemase enzyme, BLA-NDM, BLA-VIM, and BLA-IMP, in order to detect class B carbapenemase enzyme, and BLA-OXA48 in order to detect class D carbapenemase enzyme. And these are the PCR conditions. After that, all positive genes were performed the DNA sequencing, and the results were aligned in the BLAST websites in order to know the gene variants. For the result, we found that 90.5% of the isolates were positive for class B carbapenemase enzyme, including the BLA-NDM1 in 18 isolates, BLA-NDM4 in 1 isolate, and BLA-VIM2 in 1 isolate. So the BLA-NDM positive capsular pneumonia isolates have been reported in many countries around the world. In addition, we didn't find the BLA-KPC and in any isolates which is concordant with the phenotypic detections. And we also didn't find the BLA-IMP genes in any isolates. However, the occurrence of BLA-KPC and BLA-IMP genes in Thailand were 12.1% and 1.9% in central and northeastern parts of Thailand. And the 15 of these isolates were positive for the BLA-OXA48 genes. And normally the black oxa gene is originated from the Acintobacter species, and the black oxa 48 gene was firstly identified in the Capsula pneumoniae and now currently widespread in other species in Enterobacteriaceae. Moreover, one isolate carry both BLA-NDM1 and BLA-VIM2 genes, and 14 isolates carry both BLA-NDM1 and BLA-OXA48 genes. So these findings might be indicated that it's what possible that the BLA-NDM it was possible that the BLA-NDM1 and BLA-OXA48 genes might located on the same plasmid and were transferred together, causing the carbapenem resistance among the gram-negative bacteria. And we also detected the ESBL and MC productions. First, for the ESBL productions were detected by the combined like a previous method, but we used different antibiotics and inhibitors. We found that 3 isolates can produce the ESBL enzyme. And the ESBL genes were detected by multiplex PCR using this primer and this PCR condition. So the result, we found that all 21 CRKP isolates were positive for BLA-HHV and BLA-CTXM genes, and 16 isolates were positive for BLA-TEM gene. And the MC beta-lactamase productions were detected by the combined test also. And we found only one isolate produced the MC beta-lactamase enzyme. And the MC genes was also detected by the PCR method using this primer and PCR condition. And we found that 20 of these isolates were positive for MC gene. So I'm going to summarize the characteristics of the CRKP isolates. In 21 CRKP isolates, 18 isolates produce the class B carbapenemase enzyme, and most of them co-present the BLA-NDM1 with the BLA-OXA48 genes. Interestingly, another 3 isolates did not produce any carbapenemases, but they were resistant to carbapenem by other mechanisms. For the KP23s, resist to carbapenem by the presence of OXA48 gene and the MC gene and MC enzyme, while the KP31 is resist to the carbapenem by the presence of OXA48 and the ESBL productions. However, the KP16 produce only the ESBL, but according to these principles, you can see that either ESBL or DMC cannot cause the carbapenem resistance. So we need to more investigate the loss of Hollins and efflux pump in these isolates. In addition, the KP23 that I mentioned before, which carry the BLA-NDM1 genes, cannot produce the NDM1 enzyme by the diffusion methods. Is what interesting to investigate the insertion sequence here, which is the ISBA125, which provide the -35 regions of promoters for the expressions of BLA-NDM1 genes. It might be truncated in these promoters and leading to no expressions of this enzyme. For most of the CRKP isolate, as you know, that is carry both BLA-ESBL or BLA-MC genes, but they didn't produce the ESBL or MC enzyme by the diffusion method. So it might be explained that by many reasons actually, but the first reason was that either ESBL or MC genes might located on the same on the low copy number plasmid or other mobile genetic elements that not assembly to the chromosomal DNA and might result in less or no expression of either ESBL or MC enzyme. And furthermore, for the MC production, actually the MC promoters have been reported to be important for MC expressions. So if there is mutation in the promoters, it might be decrease the MC production in these isolates. That I already mentioned before, we need to investigate the proline loss and efflux pump in the KP16 isolates. The proline loss was detected by the SDS page. First, we did the sonication and added the sodium lauryl sarcosinase to destroy the inner membrane protein and ultracentrifuge it to get the outer membrane protein, or we call the OMP. Then the SDS test was performed to detect the OMPK35 and OMPK36 purines. So in the result, interestingly, we found that KP16 loss of the OMPK35 purine when it was compared with the capsular pneumonia ATCC 700603. For the overexpressions of efflux pump, it was performed by the MIC of meropenem with and without CCCP, which is an inhibitor of efflux pump by broth microdilution. It was interpreted as a positive if the MIC of meropenem alone is more than MIC of meropenem plus CCCP around 4-fold dilution. And the results show that it does not have the overexpressions of efflux pump in the KP16 strain. So I would like to summarize again. In 21 CRKP isolates, 18 isolates produce the class B carbapenemase enzyme, while 2 isolates produce either MC or ESBL enzyme with the oxalate, and 1 isolate produce the ESBL combined with the OMPK35 proline loss. So let's move to the last experiment. We study the synergistic activities of fosfomycin in combination with other antimicrobial agents against the CRKP isolates. It was performed by the checkerboard assay. Briefly, the bacterial culture was adjusted to 0.5 McFarlane standard by the sterile normal saline solution and diluted 1:100. Then they were pipetted into the 96 well plates, which contained the Muller-Hinton broth, glucose 6-phosphate, fosfomycin, and other antimicrobial agent at zero concentrations. And after that, they were incubated for 16 to 24 to 20 hours. And the fractional inhibitory concentrations index, or FICIs, were calculated by this formula and interpreted by this criteria. So we found that the synergistic effects were seen for the fosfomycin plus gentamicin in 61.9% of the isolates, followed by the fosfomycin plus imipenem and fosfomycin plus dolipenem in 47.6% of the isolates, and the fosfomycin plus levofloxacin and fosfomycin plus dipicycline in 33.3% of the isolates. And the results suggested that the fosfomycin can be used in combination with gentamicin in order to increase their activities for the treatment of CRKP infections. And additionally, it also can be prevented the emergence of fosfomycin resistance during therapy. Nevertheless, Samunitz et al. Has been reported that the percentage of the synergistic effects of fosfomycin in combination with other antimicrobial agents against the serine or the class A carbapenemase producing capsular pneumonia were higher than the strain in our study, which can produce the class B carbapenemases. And however, an antagonistic effect were observed with the fosfomycin plus meropenem, doripenem, or gentamicin again, and in a few isolates. In addition, in this publication, which was studied in the Pseudomonas aeruginosa, isolated from hospitalized patients in our hospital, actually the Songkhanakarin hospitals in Thailand, also reported the antagonistic effects that were seen for the combinations of fosfomycin with gentamicin in 27.3% and fosfomycin plus imipenem in 7% of the isolates, respectively. So for the conclusion, we got 35 Capsula pneumoniae isolates, and 21 isolates were classified as the CRKP strain with a high MSC 50 and MSC 90 value. And the mechanism of the carbapenem resistance in these isolates were class B carbapenemase in 18 isolates, ESBL plus oxafortiate in one isolate, MC plus oxafortiate one isolate, ESBL plus OMPK 35 polyns in one isolate. For the investigation of carbapenemase genes, most of the CRKP isolates harbor the BAR-NDM1 genes, and the combinations of fosfomycin and gentamicin show the highest synergistic activities among the CRKP isolates. So finally, these data could be beneficial in guiding clinical use in the future. So we would like to acknowledge Associate Professor Dr. Saranyu Shusi and Dr. Ratanaruchi Pumiset, also all of my co-authors and all scientists, staffs, and friends in divisions of biomedical science and biomedical engineering and division of internal medicine, faculty of medicine, Prince of Songkhla University, and all scientists, staff, and friends in the department of microbiology, faculty of science in Prince of Songkhla University. And finally, I would like to thank the graduate scholarship from faculty of medicine. Thank you very much for your attention.
Thank you, Arnold sir, for your great presentation. I hope all the researchers and students attending the conference will get motivations for the further activities. Once again, thank you, sir. Now, moving towards our next session. Let's continue with our second speaker. So, I, Dr. Shilpa Bhade, have a great pleasure to introduce our pre-conference second speaker, who is going to talk us about effects of ethanolic extracts of leaf, seed, and fruit of datura on kidney functions of male albino rats. For our speaker, after completion of Bachelor and Masters of Science in degree of biochemistry, sir has completed his Doctor of Philosophy degree in medical biochemistry. Currently, he is working as a senior lecturer, Department of Biochemistry at Federal University, Taraba State, Nigeria. Visiting lecturer, Department of Biological Sciences, Wakuri, Taraba State, Nigeria. Then instructional tutorial facilitator and project supervisor, Faculty of Science, National Open University, Nigeria. Sir has also very much other responsibilities in the way administrative procedures, such as Sir is a seminar coordinator and he is a secretary of academic staff union of universities at Federal University, Bakure, Nigeria. Sir is a faculty of Pure and Applied Sciences desk officer, and also he is deputy director at Center of Biotechnology, Federal University, Bakure, Nigeria. In his academic performances, he has published 60 papers in high reputed and impact factor journals. He attended 12 international conferences and 5 international and continental workshops. He attended 3 seminars as a presenter. Overall, He has very much some important professional memberships in member of Nigerian Society of Biochemistry and Molecular Biology. He is a member of Society of Basic Clinical Toxicology Nigeria. He is senior member of Asian Pacific Chemical Biological Environmental Engineering Society. He is also a member of Society of and member of Asian Council of Science Editors. He has non-academic two books published. His hobbies are reading, sports, and music. So now you all must be curious about who is today's speaker. So let's me introduce our today's speaker, Mr. Chinoru Imo sir. On the behalf of second ICTM organizers and SMBT Educational Trust, I welcome you, sir, and request you to grace us with your important views regarding the topic. Imo sir.
Right. Thank you very much. Good morning, everyone. I hope you can hear me.
You are audible,
All
sir.
right. All right. My name is Chinedu Imo, and my presentation is on the effects of ethanolic extracts of leaves, seed, and the fruit of Datura metal on kidney and liver function in male albino rats. Datura metal is a popular plant that is found in Africa and some parts of Asia. In fact, currently we know that most countries are gradually applying traditional medicine system in the curation of various diseases. And the plant itself is commonly known as devil's apple, while some refer to it as thorn apple. Others refer to it as angel's trumpet. And like I said, in Nigeria, since our major focus, the research we carried out was in Nigeria, there are three major ethnic groups in Nigeria, or three major languages. So in Igbo, it is called Miaramo. In Hausa, it is called Zakami. And in Yoruba, it is called Apikan. And the seed grows up to 200 centimeter in height, and with the fruits that bears many seeds. It is reported that each fruit bears up to 300 or more seeds, especially when it is mature and ripened. It usually open such that you can see the seeds, and the seeds are small in size. In many parts of Nigeria, the plant Datura metal is used for various purposes. In fact, in my neighborhood here, I know they cultivated it and they use it for their poultry business. Some people use the leaf, some use the stem, some use the fruit itself, while some use the seeds. Each part of it is used for various purposes. But one common thing that is known about Datura metal is that it is a potent poison and is also used for its psychoactive activities. It has been reported that by users of the plant, that once you consume it, either its extracts or in raw form, it has psychoactive activities. In fact, the questionnaire we used during the course of this research work, we discovered from respondents, those that have experienced its use previously, experienced some toxic effects, such as delirium effects. Some were able to sleep for a long time. The, the other part of it is that some that took these plant parts were able to work for a long period of time without being tired. In fact, these days, most youths make use of this plant as alternative to marijuana or to cocaine. especially when they want to, these quotes, when they want to get high. It is observed that even most people don't know that the plants have its psychoactive effects. In fact, in Nigeria, there is a tribe called Fulani. The Fulani, they are known. There is a ceremony or an event they usually celebrate or have, which some refer to as Siro. During the course of this research, we discovered that it has been reported that during that event, some of them, they use these plants to prepare concussion they use to people that represent them during a wrestling contest or other contests. In fact, in Jukun Kingdom, I discovered that where the Federal University Bukari is located. In the course of this research, I discovered that on rare occasions that datura metal is also used in preparation of certain concussions that they use on their warriors. So datura metal, when consumed, can make one to be energetic, such that you can walk for almost throughout the day without being tired. But it is temporal, and that is totally discouraged. In fact, one of my students, I think her cousin, when we were carrying out this research, we discovered that one of the cousins of my students took this plant material. After taking it, he felt, much energy in his system, and he went to the farm. He was cultivating the farm from morning till afternoon, and they were looking for him until somebody told them that they saw him in the farm. And then they went there, discovered that this young man was working with much energy and could not stop. They had to bundle him to the house and tie him with rope. Later on, they discovered that he took the seeds of datura metal. Most youths these days are beginning to abuse these plant materials, while some use the extract for some medicinal purposes, such as treating painful tumors, scabies, eczema, and then some smoke it too to relieve spasmodic asthma. So since there are some positive effects of the plant material, such as anti-asthmatic, hallucinogenic, anti-spasmodic, and bronchodilator effect, dilative effect, most people now abuse it. So our interest in this research was now to evaluate, while some people are taking these various plant material, plant parts, what are the effects on selected organs of the body in the body system. We took a close look at the kidney effects, that is its effect on kidney function. And then in other research, we looked at liver function, we looked at the lipid profile effect, and then we looked at its effect on hematological parameters. And then we discovered some positive effects and some negative effects. And in this presentation, we will concentrate more on the kidney effects. So if you usually consume this plant material, then these research findings may be very informative for you. Although some that use it in their poultry businesses use it because they believe that it helps the chickens and all the rest of them to develop immunity to withstand certain disease conditions. It can help them to treat miles and then different types of cough. This is the plant materials, part of the ones we harvested during the course of this research work. As you can see, this is, you can see the fruits. These are the fruits that is oval in shape with a spine-like epicap. And then you can see part of the leaves. And then you can see one of the fruits that we opened. When it is dry, they will start opening like this, such that you will see the arrangement of the seeds. So some people use only the seed, some use the leaf, while some use the fruits as it is. Probably you may grind it and then extract. Why some harvest it this way, then soak it in any alcoholic solvent, and then probably take it, believing that it will extract some of the active ingredients in it. Then this is another view of a mature leaf of the plant. And like I stated, this research study is aimed at presenting investigating and presenting the effects of the ethanolic leaf extract of these three parts of Datura metal on kidney function. And we believe that the result will help to reveal and then establish the likely renal function effects on consumers of Datura metal. We harvested the plant parts, that is the leaf, seed, and the fruits, from a place called Wapanyako in Okare, Taraba State, Nigeria. The plant materials were processed, they were rinsed, dried, and then we milled the leaves, the seeds, and the fruits separately into a powdered form. Known weights of each of the three plant parts were extracted using 70% ethanol by cold maceration with occasional shaking. Then, after the 48 hours maceration, each of the samples were filtered and then evaporated to dryness using a rotary evaporator. And then the extracts were then concentrated and used for the experiment. We used the 35 healthy male albino rats for this research. They were 8 weeks of age during the course of the research. The rats were purchased from a place known as VOM, is a research institute, and then transported to my department in the animal house, that is Department of Biochemistry in Federal University of Okare, Nigeria. Before the administration of the various extracts, we allowed the animals to stay for 7 days to acclimatize to the change in environment. And then all the animals had free access to commercial rat feed ad libitum. This is the experimental design. Like I said, we had 7 groups. This table represents the distribution of the animals and the administration of the plant material. Group 1 served as normal control, so they were not administered any of the plant parts of Datura metil. Group 2 and Group 3 were administered the leaf extracts of Datura metil. As you can see, Group 2, we administered a low dose, that is 300 milligram per kg body weight. So a rat that is weighing 1 kg is administered 300 milligram. Then Group 2, we administered 600 milligram of the leaf extract. Group 3, we administered 600 milligram of the leaf extract. While Group 4 and Group 5, we administered seed extract, just like the low dose for Group 4 and the high dose for Group 5. Group 6 and Group 7, we administered fruit extracts of Datura metil, the low dose for Group 6 and the high dose for Group 7. All the experimental groups, that is from Group 2 animals to Group 7 animals, received the extract for 7 consecutive days. Then the extract were administered through oral route, after which the animals were sacrificed and then blood sample collected from each of the animals in the 7 groups. The blood samples were collected and then processed to get the serum. We make use of the serum for the kidney function test and for the liver function test. But for the hematological analysis, we used the whole blood for the hematological analysis. After the blood collection, the selected organs like the kidney, the liver, the heart were harvested for histological analysis. We made use of an auto-analyzer, Landwin LWE60B, for the determination of some selected kidney function parameters. And then, after the biochemical analysis, we carried out statistical analysis to compare the differences that may have existed among the groups. These are the some of the results from the study. Uh, Table 1 shows you concentration of urea and creatinine in the rats administered various extracts, various plant part extract of datura methyl. As you can see, group 1 is the control group. If you compare the result of urea with other groups, you will discover that urea level increased in the two groups administered the leaf extract. Urea level decreased in the two groups administered the seed extract, and also decreased in the two groups administered the fruit extract. This implied that there was retention of urea in the blood in the group administered the leaf extract, while there were much excretion of urea from the blood when you compare the normal control and the group that were administered the seed extract and the fruit extract. Although none of the differences that existed, both the increase and decrease, none of them were statistically significant. However, we believe that this is a form of alteration and definitely will have an effect on the rate at which the kidney handles urea. Recall that urea is like the end metabolism west from protein metabolism from the system. So this gives an idea of how urea is being eliminated in the animals that we administered this seed extract, giving us a kind of clue what may be happening to individuals that consume this. Also, creatinine is a good indices used in evaluating renal function. Various scholars and research have reported that alteration in the level of urea and creatinine could show how the kidney is functioning. So any alteration could lead to an alteration in the renal functions. Any alteration in the levels of creatinine and urea could be a significant of the alteration of renal functions. So if you compare the results of the creatinine among the 7 groups, you will discover that the group that received the seed extract and the group that received the fruit extract costs showed the high retention of creatinine in the blood. And this is not a good condition. So it showed that while some consumers use these plant parts in the treatment of certain ailments, it is also possible that it may be causing certain toxic effects in the kidney, especially as you can see the result of the group that received 600 milligram per kg of fruit extract is 91.9 millimole per liter compared to 63.1 millimole per liter of the normal group. So you can see, although there are also increase in some other groups like group 2 that received the leaf extract, group 3 that received leaf extract, there were increase in the serum creatinine levels, although it is not significant. But this can cause alterations too, and it shows nephrotoxicity in the groups that we administer these plant extracts. So we discovered that these plant part has toxic effects on the kidney, especially when it is used continuously the way we used it in this experiment for 7 consecutive days. So it means that for this duration of time, it is possible that the plant materials may alter creatinine levels or the creatinine excretion rates or clearance rate. Definitely, it shows that the glomerulus may be influenced the rate at which they function in the animals. Now, we also looked at uh, Let me go to the next slide. Okay. We also looked at the concentrations of selected electrolytes. And we discovered that the level of sodium in all the animals were not significantly altered if you compare it with the normal control. The level of potassium were not significantly altered, although 600 milligram of the leaf extract caused a much increase, as you can see, from 4.43 millimole per liter to 5.45 millimole per liter. Although this alteration is not significant, but we know that alteration of the serum electrolytes could show cause imbalance in the transmission of fluids or the way membrane channels or membranes function, especially the heart, the liver, the kidney. And in this case, this mild alteration that was recorded shows that the extract may not actually significantly influence the membrane functionalities. However, as we can see, chloride increased from 104.75 millimole per liter to 107.75 millimole per liter in the group that received the seed extract. So mainly taking a look at this, you can see probably the seed extract and the food extract and the fruit extract actually changed the balance of the electrolytes compared to others. However, like we stated, the electrolyte balance change was not significant. Then we looked at the histological states of the kidneys of the various animals in all the groups. This photomicrograph represents the kidney cortex and the medulla of the animals in group 1, that is the normal control. So this is the normal tissue, the normal histoarchitecture of the kidney section. That is that of the group 1. So you can see the distal convoluted tubules, normal distal convoluted tubules, normal Bowman's space, and the normal Bowman's capsule, proximal convoluted tubules, and then other convoluted tubules in this second plate. So as you can see, these are the normal tissues. Then we compared it with the histological status, that is the photomicrograph of the kidney sections of other groups. So let's take a look at this. This is the photomicrograph of the second group that were administered the low dose of the leaf extract, that is 300 milligram per body kg of the leaf extract. Then if you look at this photomicrograph and compare it with group 1, you will observe that we have, it shows some sections, like you can see, I use the red arrow here to show the urinary space that increased a bit. Take a look at the red arrow, you see that is a glomerular, a kind of glomerular collapse. That is the CG you are seeing in the photograph. Okay, this glomeruli, and this is the Bowman space, and the Bowman space increased compared to the normal control. And then we observed that some tubules appears dilated. Some tubules appears dilated, as you can see. The magnification we used here is we compared it using times 100 and times 400, and then the observations were the same. So in this group, we observed increase in urinary space, and then some tubules that appeared dilated, which was not so in the normal control. So this shows that, okay, let's take a look at the third group. This photomicrograph represents group 3 that we administered 600 milligram per kg body weight of the leaf extract. And in this photomicrograph, you will observe the glomeruli in the cortical region that appeared collapsed with evidence of increased urinary space. In fact, the urinary space increase here is more than the group that received the low dose compared to the normal control. And you can also see some evidence of vacuolation in the epithelial linings of most of the tubules here. So this shows that consumption of the plant, the leaf extract in high dose for a period of 7 consecutive days could influence certain kidney functions. This is the photomicrograph of the group that received the low dose of the seed extract. Just like that of the leaf extract, there are evidence of marked glomerular collapse and increased urinary space, just like in the leaf extract. However, there is no obvious tissue morphological alteration on the cortical and the medulla tubuli. However, the alteration of the glomeruli or the glomerulus shows that it will influence the rate at which the kidney handles filtration processes. Probably the reason why the urea level and the creatinine levels were altered. Then, this is the photomicrograph of the cortex and medulla of the group that were administered 600 milligram of the seed extract. In this plate, you will observe the glomeruli and the tubules appear fairly intact with no much obvious histopathological alterations seen. So in the group that received 600 milligram, we didn't observe much histomorphological alterations. However, the low dose of the seed extract caused much damage. This group, these plates, represent the cortex and medulla of the group that we administered low dose of the fruit extract. Here, we observed the glomeruli collapsed. Some of the glomeruli collapsed, thereby giving room for increased urinary space. Although there was no obvious histopathological alterations in some of the tubules, but the alteration of the glomeruli and the Bowman's space shows that there are alterations, and there could be alterations in the way the kidney functions. And then, lastly, this is the photomicrograph of the cortex and the medulla of the group that received 600 milligram of the fruit extract. As you can see here in the cortical region, the glomeruli are enlarged, resulting to a taut adhesion to the Bowman's capsule. Again, there are inflammatory cellular infiltrations that are observed in some of the regions, as you can see in the first, the picture up there, in the periportal regions. These regions, you can see some adhesions and some infiltrations. And these could adversely alter the kidney functions, thereby influencing the levels of some of the parameters used in assessing kidney function. We observed generally in this study, and we advised that those that consume these plant parts for medicinal purposes, especially in vivo, those that use it in the system and probably not external, because a lot of people use it externally. But for those that consume it either by drinking or consuming the extracts or smoking it, should apply caution, especially the seed and fruit extracts. However, we observed in the hematological analysis that the blood cells were not negatively altered. So it doesn't have much negative influence on red blood cell, hemoglobin level, packed cell volume. In fact, the groups that we administered the plant parts showed increase in red blood cells and then increase in PCV. So it means that on certain medical conditions, the plant parts may be used for medicinal purposes. However, we observed an increase in the white blood cell levels of the group that received almost all the extract. Although it showed that the animals, the cells may have recognized the constituents of the extract as foreign material. Again, like I stated, we compared its effects on liver functions too, and discovered that some of the extracts can influence certain liver function parameters. We also looked at the protein levels and observed that the extract did not have any significant influence on the levels of protein. So while we encourage the use of these plant fats because of their positive effects on specific medical conditions, we also at the same time discourage the regular consumption of these plant fats for a long period duration of time, as it has the potency of influencing uh, kidney function. Thank you very much. Are you there?
Yeah.
Okay.
Good afternoon. I'm Kiran Suryamshi and I'm here to present a vote of thanks for today's webinar. I would like to thank our pre-conference speaker for enlightening us with their knowledge. The session was full of knowledge. Once again, I would like to thank Dr. Chinudu Emu for taking out time from his busy schedule. Also, I would like to thank students, teachers who join us today for this pre-conference session. Before I conclude the session, I would like to ask participants if you have any doubt regarding this pre-conference session given by Dr. Chinudu Imu, you can type your doubts in the control panel of this chat. Dear participants, if you have any doubt, then you can type over there. I think nobody have any doubt. Thank you so much for giving this nice session. Before I conclude this session, I would like to ask program chair to express his view. Dr. Yogesh Kushir.
Okay, namaste to all the delegates, national and the international delegates. No doubt is that the pre-conference is actually is arranged at the early time, and honestly speaking, the speaker, Professor Imo, just invited at the last moment. He gave the confirmation in the last night and gave the very beautiful session. Ability from the all the my organizing committee to give the special thanks to the Professor Evo. So the tomorrow session is started at 9 o'clock. You all of you attend the session, and the same Zoom link right now for the today's is applicable for the your international delegates e-oral presentation, which will get started at the 3:30 p.m. Okay, so it's a IST India time. I just continue to hand over the desk towards our just coordinator of the ICTM, second ICTM, Professor Suryoshi sir. Please continue.
Thank you so much, sir. And as I said, as sir said that this link will be continued for the e-oral presentation for the foreign participants. On behalf of ICTM 2021, I would like to thank students, teacher who join us for this pre-conference session, and I hope your presence. We are expecting your presence for tomorrow's session also. On behalf of ICTM 2021 and SMBT Educational Trust, I conclude pre-conference session tomorrow sharp at 9 p.m. 9 a.m. IST Mumbai will see you everyone for the main conference. Thank you very much.